the production of what we called haploid gametes through the process of meiosis. These were the sperm that are produced by males and eggs that are produced by females. And we also learned that when sexual reproduction occurs, these gametes come together to form a diploid zygote. The zygote is a single cell, but it goes on to produce the rest of the entire organism. The question we're going to begin to address today is, how do you get from a single-celled zygote to a complete organism? This is a very simple question to ask, but as it turns out, it's a very difficult one to answer. In fact, this question, how you go from one cell to many different cells in development, has been at the very center of the rise of modern biology from the time of Darwin's day in the mid-19th century up to the present day, where it's been actually one of the frontiers of modern biology. Now, I want you to think of the magnitude of this problem. A vertebrate animal, like ourselves, may be composed of as many as 10 to the 14th cells. That's 100 trillion different cells. Now, these different cells are all specialized, or can be specialized, into maybe hundreds of different types. I mean, not every one of those cells is different, but there are hundreds of different types of cells in your body. Each of those types of cells can have a unique role to play in the functioning of the whole organism. Furthermore, these various kinds of cells must occur in the right proportions and also be found in the right positions in an organism. Just look for yourself. Look for yourself in the mirror, just for a second. Without even knowing anything formal about anatomy, you can hardly help but marvel at the intricacy of your own structure, and what you might guess about the complex arrangement of cells, tissues, and organs that are arranged underneath your skin. What's truly marvelous about all this complex, substantial structure is that it arises from a single cell formed when a sperm and an egg fuse. Today we're going to begin a series of lectures on development. We'll begin in this lecture by first considering the general issues we need to explore, the fundamental issues that we need to address to understand how development proceeds. Our goal today will be to provide an overview of what must be involved when an organism develops, to serve as kind of a roadmap for the material that follows. Then, starting in the next lecture, we'll look in some detail at the basic patterns of development in animals and the key mechanisms involved in producing a complex multicellular organism from a single fertilized egg. As we'll see, there are many questions that still need to be answered in this very exciting and dynamic field of research. Well, development really presents us with two broad questions that we need to address, or I might say two puzzles we need to solve. The first puzzle concerns the question of diversity. All organisms, multicellular organisms, start from a single cell. And by and large, these cells that they start from all look pretty much the same. Yet, the zygotes from different species, of course, develop into radically different kinds of organisms, as diverse as oak trees and earthworms or sea urchins and giraffes. And more astonishing, as I pointed out, within each of these kinds of organisms, there are many, many different kinds of cells, each performing different functions and each occurring in different proportions and different positions in that organism. We start with a single cell of one kind and end up with as many as 1 trillion cells of hundreds of different kinds, all in the right place. So the question is this: how does this diversity, both the diversity we observe between species and, more importantly, the diversity we observe within a single species, arise from a single unformed, or single uniform, I should say, cell? Now, the second puzzle concerns the question of consistency. And this is really the flip side of the coin. Each time the zygote of a particular species develops into an organism, it comes up with more or less the same result every time. That's to say, things go wrong in development relatively rarely. In spite of all of this complexity, an organism more or less will turn out the same each time. Now, of course, each individual of a particular species will be slightly different, but what's more important and more remarkable, really, is that they're all mostly the same. So this leads us really to the second question, which is, how is this consistency in development maintained? That is, what ensures that we get the same outcome each time? The answers to both of these questions that is, how we get diversity and how we get consistency, must involve some set of developmental mechanisms. So the most fundamental question we need to address is this: what dictates the way that single cell can turn into many different kinds of cells, all in the right place, in the right numbers? Actually, it was the Greek philosopher Aristotle who contributed much to our modern thinking in biology, who actually originally proposed that organisms must develop from a uniform single cell, a cell that has no structure. We refer to this idea today as epigenesis. Now, epigenesis implies that there must be some way to create diversity from something that has no inherent structure. Aristotle himself wasn't particularly concerned with the details of how that could occur because he was just sort of interested in the philosophy of this. So the complexities weren't really a problem, but there are complexities that we need to consider. And in fact, later on, people began to ask, how is this possible? How can you get an organism coming out of a single cell? An alternative view we call preformation. Now, the view preformation suggests that, in fact, an egg cell contains within it an entire miniature organism, and that what happens in development is that this organism just expands out. Now, the idea of preformation is in some ways easier to accept because it doesn't require us to posit any special mechanisms or mysterious forces that create diverse parts out of a uniform start. Of course, preformation has its own problems, which is to figure out how you can actually fit an entire miniature organism into a single cell. Actually, early on, really before the 18th century or so, that wasn't much of a problem for scientists because they didn't understand the structure of cells. It was really only in the 19th century when what we call today cell theory arose. Cell theory being the idea that all living organisms are composed of discrete cells. With the advent of cell theory, people started looking in more detail at the nature of those cells. And with improved microscopes, it became clear that if you looked very closely at a single zygote, or the earliest phases of a developing organism, you didn't see any miniature preformed organism enclosed in that cell. For example, in the 1760s, a famous German microscopist named Caspar Friedrich Wolff described the development of chick embryos in some detail. And he was able to show that parts of the chick, for example, the intestine, would emerge de novo from nothing out of other parts of the organism, of the developing embryo, that didn't have any structure and that, in fact, weren't found in the adult anywhere. So, for example, a sheet of tissue that was a uniform sheet would bend around and close in on itself, Wolff saw, to form the gut. And so this kind of evidence showed that somehow structure did arise as organisms developed. And it was this kind of evidence that ultimately ruled out the theory of preformation. Now, I'll add that preformationists hung on for quite some time because they could always claim that you didn't really describe the structure in enough detail to see what must have been there. The microscope wasn't really good enough to be able to see the organism that must be enclosed. But by the time that the 19th century moved on, most people realized from detailed embryological studies being studies of different kinds of organisms as they're developing by a group of scientists that we would call embryologists. They realized from these studies that the emergence of structure must somehow involve mechanisms to create new structures where none existed before, not to simply unfold pre-existing structures, as the preformationists would argue. Of course, the acceptance of this epigenetic hypothesis brought the fundamental question of development into focus. And that is, as we said before, how can a single uniform cell turn into many different kinds of cells? Having argued in favor of epigenesis, for example, embryologists such as Wolff could only propose that there must be some unique fundamental force of nature. Actually, Wolff called this force in English translation the essential force, which is a pretty vague term. And he argued that this must be some natural force that might be analogous to a natural force like gravity or magnetism, which back then weren't completely understood. Just a force that you knew existed because it acted on nature. And in this case, the essential force would be some unknown or undescribed force that was acting on developing organisms to create structure. As I said, a pretty vague idea. Now, as biologists in the later part of the 19th century, in the early part of the 20th century, began to understand in more detail how cells worked, that is, as cell biology began to develop as a science. And especially as they began to understand how cells divided through the process of mitosis, and also began to understand the fundamentals of genetics, the idea of some vague essential force was quickly replaced by proposals for specific mechanisms that could account both for the diversity and consistency of development. So what I want to do now is to consider what these mechanisms could be. Or to put it another way, I want to ask what are the factors that could possibly control the development of an organism? Well, given what we've learned so far earlier in the course, we might think that the answer to this question is pretty simple. And that is that development must be controlled by genes. Well, the earliest embryologists didn't know anything about genes, of course, or really understand genetics. But we know today that different zygotes of different species must have different genes. They have different genomes. And so it seems pretty reasonable to us, at least, that differences in these genomes must be responsible in some way for the diversity of development of different species, and further, that an organism's genes are what directs development so that a particular species always comes out the same way. The emergence of structure must have something to do with the information stored in the genome. That can't be disputed. Genes code for proteins, as we've learned. Proteins are what determine the structure and function of a cell, and therefore you could imagine that the proteins coded for these genes must determine the way that cells come out in different patterns. But this has to be an incomplete answer. As we'll see, genes by themselves can't account for all aspects of development. Why not? Why is it obvious that genes alone can't determine the outcome of development? Well, there are many reasons that could be given, but the most obvious one comes from the observation that the development of multicellular organisms doesn't simply lead to a large group of identical cells. The single cell we start out with has a given set of genes. And when cells divide, we know from our earlier discussions that their genes are accurately copied and transmitted to the daughter cells. If genes were all that were responsible for controlling development, then why wouldn't every daughter cell produced as a zygote develops into many cells, why wouldn't all of these daughter cells be exactly the same, given that they all have identical sets of genes? Well, okay, you'd say we understand also from a few lectures ago that genes can be turned on and off. In other words, different cells can express different genes. But the question then just moves a step backwards, and it becomes what ultimately is responsible for determining which genes in a cell are expressed. Clearly, something other than the genes themselves must be responsible for the differences, I should say, something other than the genes in a particular cell must be responsible for the differences that arise among cells in the development of multicellular organisms. That is, there has to be something that's external to that zygote's genome that must determine the pattern in which genes are turned on and off. So we can refer to the factors that could turn genes on and off in general as the environment in which the genes find themselves. So what I want to do now is to turn to the question of what do we mean by the environment? What is it that could affect how genes are turned on and off? We're going to consider this question in general terms first in order to provide an outline of the issues we're going to look in more detail as we go along. Of course, the expression of genes can be modified or modulated by environmental factors that are completely external to the developing organism. And by environment here, I mean things like temperature or nutrition or other external outside things, the kinds of things that we would initially think of as the environment. And actually, the external environment can have an extraordinarily profound effect on the outcome of development in some cases. I want to give you a fairly extreme example just to show you how radically this external environment can affect the development of the organism. This is an example that comes from looking at the development of moths and butterflies. It turns out that some species of moths and butterflies develop into very different looking caterpillars or adults. The caterpillars are the young larval form, as you know, depending on the time of year that they happen to be born. In other words, a particular species of butterfly or moth might end up looking different depending on whether it's born in the spring or the summer. In fact, they can look so different that these two different forms, or morphs, would have been initially considered two different species. The best example I know of that illustrates this involves seasonal change in morphology in a moth with the scientific name Nemoria arizonaria, which has been studied by Eric Green at the University of Montana. The caterpillars of this species are camouflaged from predators by resembling the substrates they sit on. This is what we would call mimicry, and it's a very common adaptation among insects for avoiding visual predators. They look like what they're sitting on, the predator can't see them. Now, in the spring, caterpillars eat the catkins, or that's what the name is for the little group of flowers that are produced by oak trees. The caterpillars also mimic these catkins remarkably well. If you looked at an oak tree and it had a nemoria on it, you might actually mistake one of the caterpillars for a little flowering stalk. It's a remarkable mimicry. Later in the summer, however, this same species of caterpillar looks completely different. Instead of looking like the catkin of the oak tree, it looks like a little stem or branch. Again, quite remarkably well camouflaged. Now, this is adaptive because early on in the spring, when the trees are flowering, they're covered with catkins. But in the summer, the catkins are gone. And so a caterpillar that looked like a catkin would be standing out. Well, Eric Green was able to show that it's what the caterpillar eats that turns it into one form or another. And specifically, he was able to show that early on in the spring, the caterpillars are actually eating the catkins. But later, when the catkins are gone, they start eating oak leaves. And there's a chemical in the oak leaves that changes the developmental program of the caterpillar and ends up giving it a completely different morphology. Now, in most cases, the external environment has a much less profound effect on the development of an organism, but these cases of what we would call adaptive phenotypic plasticity make the case well that environmental factors can have a profound effect in general on the way that genes influence development. Now, there's two other kinds of what I'll call, in quotes, environmental factors that also can express the genes of a developing organism. Unlike the external environments, like the chemicals that these caterpillars were eating or temperature and so forth, these environmental factors refer to things that are actually internal to the organism, even though they're external to the organism's genes. The first crucial set of environmental factors that plays a central role in development are the cellular contents of the fertilized egg. That is, the contents of the female gamete. As we'll see in more detail later, the control of the earliest stages of development in many animals is exerted almost entirely by the existing contents of the egg that gets fertilized, not the genes that are found in that egg. These contents that are put in by the mother include an extensive amount of messenger RNA, that is, transcription of the mother's DNA that is then put into the egg, and also a number of proteins, again, produced by the mother and put into the egg. These stored factors that are contributed by the mother we call cytoplasmic determinants because they will determine the behavior of that cell. I'll also call them maternal determinants from time to time because they're necessarily from the mother. We'll see how it's the mother's genome, not the offspring's own genes, that control the first few critical steps in development. And in fact, at this time, the genome of the zygote itself plays little or no role. Now, the second set of crucial environmental factors, environment internal to the organism, that influence development comes from interactions among cells. This kind of cell-cell interaction, involving the kind of cellular signaling that we discussed just a few lectures ago, becomes especially important in later phases of development, as cells become increasingly specialized and as they assume their proper positions and numbers in the maturing organism. As a growing organism begins to develop different kinds of cells in different positions, these cells will start signaling to each other, and a cell will respond to these signals depending on where it finds itself relative to the other cells. In other words, it might get a certain set of signals if it's in one place in the organism because the cells around it are sending that particular signal, but if the cell finds itself in a different place in the developing organism, it'll get a different set of signals because it's surrounded by different kinds of cells. In this way, cells begin to influence each other's developmental fate, or to put it another way, the adjacent cells to a developing cell will be sending signals that will determine which genes get turned on and off in that particular cell. The take-home message here is that the emergence of a complex organism through development is not just due to the genetic blueprint, that is, the genes found in the organism's own genome, but also to the environment that those genes find themselves that influence the expression of that blueprint. And this environment not only includes what we would normally think of as sort of the external environment, such as temperature or other things that the organism finds itself developing within, it also includes the intracellular environment that the cell's DNA finds itself in. Those are the cytoplasmic determinants that are packed into the zygote by the mother, and also the extracellular environment formed by other cells surrounding a developing cell. Well, now that we have a framework for thinking about the factors that control development, let's next outline what it is that needs to be controlled. In other words, what are the kinds of mechanisms that we're going to have to consider in order to understand the development of a single cell into countless different cells in different kinds and different places, of different kinds and different places? Now, again, if you think of a complex organism such as ourselves or the caterpillar I discussed earlier, you might think that there must be an extensive list of mechanisms that are responsible for coming up with all of this complex, diverse cell types and morphology. But in fact, and fortunately for us, we can boil the answer down into just sort of three main sets of processes. So let me list those for you now. The first process is simply cell division. That is, when and how often cells divide. We've already looked at this mechanism earlier in the course when we studied mitosis. Of course, it's obvious that in order to get from a single cell to the many cells that make up an organism, cells have to divide, and they have to do so in the proper amounts. It turns out that when and where cells divide play a major role in the emergence of the structure of the organism. The second process we need to consider is called differentiation. Now, differentiation is the process by which cells become specialized to have specific structures and functions. The process of differentiation is where differences in gene expression most obviously play an essential role. And so it's here where our understanding of gene regulation will come into play. We'll see later how the process by which a cell's eventual fate is determined is a gradual one, with cells early on in development having the potential to differentiate into all sorts of different kinds of cells, but eventually having this potential diminish in later stages of development. The cells become more and more what we'll call determined and eventually differentiate into a particular kind of cell. Now, the third main process of development we'll call morphogenesis. Morphogenesis literally means creation of form. In development, morphogenesis refers to the mechanisms responsible for changes in shape and organization of the structure of organisms once they get into a stage where there are a lot of cells. There are a number of mechanisms involved in morphogenesis, and let me just list a few of the most important ones. One very important mechanism in morphogenesis involves the cells themselves changing their shape. Imagine for a moment that you're looking edge-on at a single sheet of cells, okay, a one cell layer thick sheet of cells. And because you're looking at it edge-on, you might imagine that as just a single row of cells, and because you're looking at the edge. And imagine that all of those cells just happen to be all rectangular. So you have a flat sheet with rectangular cells all neatly lined up. Now imagine that at one point in this line of cells, the bottom part of the cells began to expand relative to the top part of the cells, so that those cells in that section became shaped more like a wedge. Now, if you think of what would happen, you'd realize that as these cells became wedge-like, the sheet would begin to distend. In fact, it would sort of bulge downward. And as it bulged downward, it might form like a little depression or groove in that sheet. In fact, you might imagine that it bulges downward enough that the sheet could actually sort of come around over and form an enclosure. This is just an example of how changes in cell shape can change the layers of an organism. And as we'll see, this is a very important mechanism of morphogenesis. Another mechanism that's very important involves the actual movement of cells. Cells move about extensively during development. They literally get up and crawl. They do this by sending out cellular protrusions that reach out and then drag the rest of the cell along behind it, much the same way that a single-celled amoeba, which you might have looked at under a microscope sometime, crawls around. Actually, there are what we call glycoproteins on the surfaces of the cell. Remember, glycoproteins, we've learned, are proteins that have had a sugar part added to them. These glycoproteins serve as cell adhesion molecules that help the cells to stick to each other as they move about. And furthermore, different cells will have different kinds of glycoproteins that recognize each other. Remember, when we talked about protein trafficking, we learned that glycoproteins can serve as membrane recognition mechanisms. So on the outside of the cell, these glycoproteins, which we generally call cell adhesion molecules, make sure that cells only attach to and stick to the right kind of cell, or a particular kind of cell. Meaning that when cells are moving about, they can use these cell adhesion molecules as essentially pathways to follow. Now, a final mechanism involved in morphogenesis that I want to mention briefly involves the fact that some cells in a developing organism are simply pre-programmed to die. This is a process that we call apoptosis, or programmed cell death. This so-called programmed cell death is important, for example, in creating the spaces between our fingers and toes. When our hand first develops, there's a continuous sheet of tissue, webbing really, that is extending between all of our fingers. But at some point in development, the cells that are in the middle get a message that tells them to simply die. And when they die, their death carves out the shape of the spaces between our fingers. Well, not surprisingly, the details of how an organism develops from an egg to the rest of a fully developed organism varies greatly from species to species. There's one more point I want to make, though, about development in general before we move on to the specifics. And that is, early on in development, the organism has to develop the basic axes of its body plan. For example, if you consider a developing plant, there is an inherent axis that we would call the root to shoot axis, the down to up axis. And the different parts of the plant develop differently according whether they're in roots or up in shoots. In animals, the most fundamental asymmetry established is along what we call the head to tail axis, or more formally, the anterior to posterior axis, which sets up the development of structures that are more head-like versus structures that are more tail-like. Well, now that we've laid out some of the general issues in development that we have to consider, we can begin to look in more detail at the mechanisms that lead to the development of complex organisms. For the sake of time, we're going to focus the remainder of our discussion of development on animals, beginning with a description of how a zygote gets transformed first into a ball of cells and then into a more complex structure. And this is the story that we'll pick up with next time.